All right, in this study we have 5 cystic fibrosis volunteers and then 5 non-cystic fibrosis volunteers. And these guys are important because this is what we're finding in lungs in general. Here we have one person with asthma and one person living with a CF individual. And the general take-home message is that when you look in cystic fibrosis versus non-cystic fibrosis, yeah, my thing is tiny. So the bottom versus the top, what you'll see is that that cystic fibrosis tends to have more viruses associated with that, and actually most of them are going to be phage of some sort, though you do see a lot of eukaryotic viruses. The total diversity is maybe 175 viruses there. However, if you have something, so you see the 2 that are in boxes and the non-CF, if you have something that's actually keeping you from clearing your lungs well, you also see a lot of viral diversity. Or we think if you're in an environment where there's high, you're exposed to a lot of viruses that are normally found in the lungs, you're inhaling them. And that's one of the people that's living with a CF person. Okay. And this is just another way of showing that. Okay, dead. So here's the non-CF over here. Then here's the CF people right here. And then you can see that people with asthma and the people living with CF tend to be getting more of that stuff in their lungs. And the general rule with more samples now, what we're finding is that it's a lot about what you're breathing in, which I guess makes sense, right? So you're just breathing it in, and that's one of the main things that you're holding in your lungs. And we're probably clearing it, whereas the cystic fibrosis people are not. Okay. However, if you look at the genes that are there, not just the types of viruses, but the genes, you start to see these really cool things. So here you have the non-CF are up here, they cluster together. These are the CF people here, and then this is this other CF person is stuck way out here. And the reason that the genes in the viruses are so different is really because of one gene in this case, and it's called REL-A, and I'll tell you about that. I'll show you this. It's right here. So REL-A is involved in regulating PPPG-PP, which is one of these central metabolism genes. It has a lot of interesting behaviors, but one of them is it increases antibiotic resistance to virulence and P. aeruginosa. What's interesting about this patient is that they died from Pseudomonas aeruginosa that was resistant to all the antibiotics that they had tried out about 3 weeks after this sample was taken, right? So we think that what you've seen is a massive expansion of REL-A through the whole system. What we know is that the REL-A gene here is in at least 5 different viruses or 5 different phage, right? So it's jumped between different phage and is being expanded through the system. All right. The other thing that we see in these guys is that there's an increase in the genes associated with anaerobic degradation of aromatic amino acids in the CF lungs, or in the viruses from the CF lungs. And what this looks like is basically we think it's a denitrified system. So this is ecology, sorry about that. So the idea being is that you've got, so here's your tissue, then this is a cross-section through one of the lung airways, and this is the microbial community. What we think is happening is that they're eating aromatic amino acids that are coming in from the tissue damage, and then they're shunting electrons back out to the blood this direction. Okay? And there's a lot of evidence pointing that direction now, which might give you some potential ways of actually killing that community right there. And this is the model that as we look at CF right now. So we think that there are basically 2 communities. Okay? The first is this one where you've got the Pseudomonas aeruginosa forming the alginate biofilms with the mucus all together. And then in the center of it are where those strip anaerobes. So I don't know if you guys know this, but in cystic fibrosis, you actually have this horrible infiltration of really nasty anaerobic bacteria, the ones that normally are associated with your teeth, problems in your teeth. And they'll form, they're hiding in there somewhere. So we think they're surrounded by these, by Pseudomonas aeruginosa, and that this is actually a slow-growing community that's essentially a climax community in ecology. right? So what this means is a slow-growing community that has a central organism that keeps it all going, in this case being P. aeruginosa, okay? And that this community is actually only able to gain space in the lung when you go through these attack communities. So these are when things like strep or staph come in, or other viruses like influenza or something. So something that clears space, causes scarring, then you get this climax community to walk up the lung. And this is kind of what it looks like. Okay, so in this case we have a patient who has the classical community living in them, and they were stable enough to get a lung transplant. So this is the lung, after the lung transplant. This is before. And what's interesting is you'll see essentially all the microbial communities are in the upper airways, right? And that's mostly because when you're coughing, right, you're not able to, you can cough deep and you can knock things out of your bottom airways much easier, but your upper airways are stagnant or more stagnant. And so that's where they're basically upper airway diseases for the most part. However, so this person gets a lung transplant. This person was an athlete that went into this horrible, so like they're doing perfect with their lung function, and then all of a sudden they go into this horrible decline, and they died within about 3 months of being an athlete, running marathons, to just falling apart. And what's happened in this case is they have basically total infiltration of their lungs by this microbial community. So it's taken over the whole system. So this would be our attack community, and this would be our climax community. And what we're doing here is, in this study, we're going to go into the lungs, and we're going to basically sterilize it and go in and then dissect out different parts of each lobe. So we're going to work at the lobe level now. And when we do this, and then we take the DNA samples and we look at these different, the different regions of the lung using 16S, it's very similar to what we saw in the gut. It's actually not that different in different places. Okay, so you see that these are the different 16S by Pyro sequencing, and essentially this whole lung looks the same. Even though we know we have more Pseudomonas up here because of the upper airway, they stray into this part, and you see basically the same community. Okay? The other one happens in this case. This is again the blue and the gray here are kind of the same microbes. They're just a little different. And in this case, you have this through the lung. The only difference you have is right in this place, right in the lower airway, which actually there was something weird going on right there. All right, so when it comes to the microbes, we don't see much difference in different regions of the lung. But when we go in and we start looking at the viral communities, we see very big differences. And this is actually one of the most weird things we've ever observed. So in those upper airways, right, so remember this is where, let me flip back. Okay, so here, we're looking right here now at the viruses. Okay? Basically, there are no viruses there. Okay? We see no phage. Okay? The only viruses that we're picking up essentially are herpes-like viruses. Okay? Even though when you look at them under the microscope, there's tons of bacteria in there. Right? So this microbial community is not functioning normally. So we don't have this turnover of the viruses versus the phage. The other thing that's happening, which I think is really, when you guys are thinking about the medical implications and how to do this, notice right here, this is actually one of the lungs, and we notice when we're looking at it that there's little cysts in the system, all right? And those cysts are basically an HPV carcinoma, right? So we didn't know at the time, and of course, when you're a CF doctor, you would never actually check someone for lung cancer, right? Because they already have lung function problems. So these guys, this person in this case actually had a carcinoma growing at the same time they're fighting the cystic fibrosis disease itself. Okay? And we find HPV, by the way, all over. HPV is just really a common thing. Okay? The other things that are going on here is that the viruses in this system are moving around some of the important phenotypes for the bacteria. One is the mucoidy phenotype of Pseudomonas aeruginosa, as well as actually a couple other gram-negative bacteria. And this is where the antibiotic resistance genes are. So there's massive number of different antibiotic resistance genes in different parts of the lung of a cystic fibrosis patient that seem to be, encoded in the viral fractions. All right, so what's actually going on here? So here we've got basically no viruses in this lung. It actually goes back to this idea of temperate behavior. So our model is that when you're so close together and you're stuck in those biofilms, phage cannot do their job of attacking, right? They can't go in, kill the bacteria, and then find a new host. So everything is a lysogen. And that's essentially what we find. So if you take these lungs, you dissect them, you get P. aeruginosa isolates, you prep the viruses, and then you do a cross-infection study. What you find is that there are no lytic viruses in the system. Okay? So only in one case do we actually find any phage that was able to attack P. aeruginosa in this one. However, all right, so then if you go in and you look for them directly, okay, so here you dissect the lung, you embed it, and then you look for the dividing cells, which tell you that the cells are growing. So we actually find a fairly high number of dividing cells. That's actually a very active community. But we find no infected cells. So normally you would find in here ones that have the capsid, the viral heads living in these cells. And we actually find none. Okay? And we suck at microscopy, I'll be the first to admit, but we do this in a whole bunch of systems, and we just don't see them in this system. So unless there's something fundamentally different, when you get those biofilms, we do not see a lot of infected cells. Okay? And then finally, the classical sort of approach to proving that you have lysogens is to do inductions with mitomycin C. And this is what it looks like if you've got no mitomycin C, the strains just grow up. If you add mitomycin C, you see them grow up, and then the viruses burst, and then you go down to no cells. And that's for 100% of the P. aeruginosa isolates we have from cystic fibrosis lungs. They have this behavior. So they're all carrying a temperate phage as far as we can. And this does give you a new treatment possibility to think about, because if they're all lysogens, we should be looking for, in the pharmacia, for different drugs that induce phage. And actually there are some some drugs that work this way, depending on the phage. Are you guys doing things like this? Does that make sense?
Yeah. Well, we are doing that anyway, because colimycin, since they all receive, is inducing.
Yeah, exactly. So you see a lot of this behavior. That's one of the main ones that we've seen this. Yeah. So this is from a treatment point of view, there might be really this is a good one to think about going down. Soy sauce is also really good, so I don't know how you get it in there, but all right. So what we've found so far is most of the unknowns, of course, are viral. Temperate viruses are more common, and that seems to be the rule for the human microbiome in general. We do actually see a lot of herpes viruses and HPV in the later ends of the disease. We don't see as many in the younger patients, okay? And I think that's because you haven't damaged the tissue as much, so you're not getting this constant induction of the endogenous viruses for us. Okay? And then the phage really, at least in the biofilms, aren't working to control the bacteria. Right? And this goes back to this other thing that I know you guys are interested in, and I just wanted to show you this because it's old, but it's still, we've been doing a lot more of it lately, and it's really fun. So we're looking for just eukaryotic viruses in human plasma for the most part. And what we do in this case is we we do the same sorts of preps, but our target